Hi everyone, I'm Namita Shroff, a staff scientist at UW-Madison in Erica Majumdar's lab, and thank you all for having me here. I am going to be talking about microbes, and there haven't been a lot of talks about microbes. So, quick poll, any microbiologists in the room? Okay, a few. That gives me an idea of what to talk about, introduction-wise. So, that picture there is a picture of our local landfill in Madison, who we collaborated with to get samples for this study. And thank you to all of you for introducing my work for me. I don't really have to talk about this. Just a couple things that I want to point out. Plastics are everywhere, and a lot of our waste gets landfilled. And 18% of the waste that the US generated in 2018 was plastic. That is 27 million tons. And as you can imagine, that is only going up. And just a quick note, like we were talking in the earlier talk, we were talking about regulations around plastics, and plastic bags are the easiest low-hanging fruit to target, right? But if you look at this graph here, a lot of plastic manufacturing is packaging. So just an idea, that's something else that we can target regulation-wise. I don't need to introduce this, so I'm going to skip this. We all know plastics have a lot of toxic effects. Okay, landfills. How do landfills work? Landfills are now municipal sanitary waste, or MSW landfills. And we touched upon this a little bit yesterday in Melissa's talk. Landfills, at least in some countries, are a huge point of contamination when it comes to all of contaminants migrating through the environment. In the US, landfills are highly engineered, but this only happened in the early 1980s. Before that, even in the US, landfills were big dumps. They may or may not have had a liner, maybe a clay liner. So a lot of contamination has happened even from landfills in the US. But current landfills, or MSW landfills, still a big hole in the ground, but they have a complex network of a liner system that prevents stuff from leaking out into the environment. And there's two things that they're concerned about leaking out. One is the trash itself that goes into the landfills. Second is leachate. Leachate is storm water or drain water that percolates through all of that trash from top to bottom, solubilizing anything it can along the way. And most landfills have leachate collection systems that collect all of that leachate, decontaminate it before sending it out for waste treatment. Okay, microbial processes within a landfill. There are a few that happen, and for that it's important to understand that landfills, being structured the way they are aerobic on the top, and then there is an oxygen gradient, anaerobic all the way at the bottom. So based on that, the processes that happen are aerobic in the top. So that's where decomposition of all of your organic matter happens. In the middle layers is where you see a lot of fermentation, anaerobic processes. And all the way at the bottom, where the temperature is nice and hot, there's a lot of moisture there. You have methanogenesis, where microbes are producing methane gas. Now, why should we understand all of this? It's important because there's a lot of microbial potential when it comes to managing landfills. Microbes can be used for recovering resources from the landfill. So for example, that methane gas that I just talked about. A lot of landfills convert that methane gas to biogas or RNG, and it's incorporated into the gas needs for the city, which is actually what happens at Dane County landfill. You can use microbes to decontaminate that leachate that I talked about. You can use microbes to degrade plastics. And eventually, all of this can lead to better landfill and land management practices. So a little introduction about microbes. Microbes are awesome. They carry out a lot of different metabolic reactions, so basically eating. They take in a lot of basic nutrients that humans do not have the ability to metabolize. And they do awesome things like make methane. They also obviously use it to grow, but you can use microbes to degrade a lot of these contaminants that we are all talking about in this conference. And what microbes are contributing to these landfill processes? This is a general overview. The aerobic processes are carried out by fungi. Fungi are great at breaking down food waste, for example. The middle layers are dominated by bacteria that carry out fermentation, and then archaea in the bottom layers, which use those fermentation products to make your methane gas. So there's a lot of teamwork going on throughout the landfill between these microbes. And there are a lot of other microbes. We studied only these 3 types in our study, but there are viruses, algae, amoeba, etc. So this was our overarching goal for the project. We wanted to characterize the landfill biogeochemistry, look for plastic degrading microbes, purify isolates from those communities of microbes, what enzymes are they using to break down the plastics, construct synthetic communities, and simulate landfill plastic degradation in the lab, and ultimately apply it to landfill characterization. So this was the landfill sampling process. This work was done by a few of the grad students in Erica's lab a few years ago. So the landfill operator basically told the students when they were going to dig, where they were digging, what depth they're digging at. So the students just stood there with a bleached bucket, and every 10 feet, the operator paused, and the trash that came out, they collected and brought it back into the lab. So this work a majority of this work was done by this grad student, Dama, in the lab. And one of the things she did with these trash samples is separated out all of the plastics and then the soil that was associated with those plastics. And you can see the plastic samples here on the top. And this is going from a shallow depth to a deeper depth. So you can see that the plastics are still pretty intact, even at 88 feet of depth. And the main takeaway point here is I want you to see there's a lot of colors, which indicates diversity in the microbial communities that you see. And also the colors are different between plastics and their associated soils. So there's a lot of different microbes. And this is only bacteria. She did the same with fungi, with archaea. I'm not going to show you that data, but we see similar trends. And what she also did was compared her data with this plastic database, which is available. It's an open source repository that is tracking any new plastic degradation literature that's coming out in terms of microbes. So again, the main takeaway point is there's a lot of genera that are common between what she got from the landfill communities and what you see here. So there are plastic degrading microbes in the landfill. Now, in microbiology, one of the things a lot of microbiologists like to do is something we call enrichments. So we mimic the environment in the lab and see if we can tease apart the different mechanisms that the microbes are using to grow. So what she did here is, in this first tube, took a few pieces of those plastic trash, added a rich medium, a rich organic complex medium, to get a lot of that biomass going. And serial transfers into rich media. Ultimately, serial transfers into minimal medium where the plastic is the sole carbon source. So the microbes are forced to use only the plastic and nothing else. And after, as you can see, 9 months of growth, Dama was able to harvest some microbes from some of the plastic communities, and these were frozen, and she studied substrate utilization from those microbial communities. So here what she did is then took pure forms of those substrates individually, and they're not mixed plastics, so she studied additives that go into plastics, short chain plastics, long chain plastics, and basically these are some of the ones that she tested. To cut a very long story short, what she saw is, yes, microbes are using short chain alkanes and additives as carbon sources. Plastics, Not so much. There is some growth, not a whole lot. Long chain plastics, no, there isn't. And just to note, this was all done in aerobic conditions to give the microbes an optimum condition for carrying out some of those plastic degradation mechanisms. These are pictures of some of the isolates from some of those individual substrate utilization tests. And the main thing that I want to point out here is even though alkanes was where we saw most growth, but the genera that we see across the board are somewhat common. So that's promising. A very cool outcome of this project was, this was done by David, another grad student who worked on this project. Caprolactam is one of the monomers that is used to make nylon. And David extracted caprolactam from one of the landfill nylon samples, and he found that some of the communities were able to grow on caprolactam as the sole source of carbon and nitrogen. And he actually purified a novel isolate from one of these communities that is definitely degrading caprolactam in his work. And there's more work around this, but that is yet to be published. So this is a great example of how microbial metabolism is probably at work in decontaminating some of the contaminants in that landfill. So in the end, what did we learn? Landfills are very heterogeneous, both in terms of trash and the microbes. There are plastic degrading microbes in the landfill, but we do not think there is a lot of plastic degradation happening in the landfill. One, we see pieces of plastic just as is surviving in the landfills. We think the conditions are not right. It's too anaerobic way down at the bottom over there. And microbes are really good at eating the easiest to eat things first. And in a landfill, there's a lot of those things. So they will eat everything else first and go to the plastic only when there's nothing else left. The good news is we think they are metabolizing some of those additives. So there is some decontamination work happening. And with that, I want to thank Erica's lab and all of the awesome people who have worked on this project. All the people in bold are major contributors to the project. This was funded by NSF and a graduate research fellowship. And with that, happy to take questions. We do have time for questions. We have one over there and one right here.
Great talk.
Thanks.
Did you all run any qPCR or single-cell RNA sequencing from freshly collected to whenever you were doing the different feeding trials?
Yeah, not yet, but that is the plan with some of those isolates that we have. We are going to be making synthetic communities and then applying them to different plastics and then doing some transcriptomics and metabolomics on those communities.
Yeah, would it be economically or practically worthwhile to change the environment down at the bottom of the landfill to enhance that plastic degradation, in the anaerobic environment?
So there are some landfills that are trying that out where they are turning over the trash. So they're introducing more oxygen into the landfills. It's not easy to do because, as you can imagine, if the landfill is 300 feet deep, that's a lot of digging up that they have to do. Some landfills are trying some other techniques of aerating the bottom of the landfills, but that is all new technology and it's not widely used yet.
Yeah, great talk. Great talk. One question, when you do the enrichment, you transferring them into the rich media, do you think you lose in some of the important microbes in there?
Yeah.
The other question is, when you have other additives in the plastic, in your enrichment, how that affects?
Yeah, great question. Yes, we think we might be losing some of the microbes in those enrichments, which is why we have all the samples frozen, all the landfill trash. We have a special landfill freezer in the lab. So we are at any point able to go back to our landfill samples and we want to test it with different medium conditions. So different media or different growth conditions to see if our microbial patterns change in those initial enrichments. And yes, the additives of those plastics are there. They might be either interfering or allowing other microbes to grow. But that's why we want to try different combinations and see what combination gives us the most diverse set of microbes that we can use.